this information, integrate, and combine final image in accordance with the integration of signals about dips, visual stimuli, light, hearing, sounds, everything. For example, optical illusions. As classic examples, optical illusions. I don't know about, maybe you can see on screen, but I can see on my screen, computer screen, moving circles. What do you see now? Moving circles or stable circles? Stable. No, this illusion doesn't work usually in screen projection, with projection. But I can explain with more simple illusion, you know. What line is longer, first or second? Second. Most people see second line as longer, first shorter, but these lines are equal. They have equal lengths. Details with arrows, arrows direction to inside or outside change your perception. It's a classic example of a simple example of optical illusion. And actually, all perceived world is illusion. You don't know about external environments, exact information. You don't have. But it's not crucial because you can use this information, perceived information, subjective information, to coordinate your behavior and to reach your life goals. This is not crucial. It can be crucial with diagnosis. For example, your patient has pain around location about some teeth. But this patient maybe that doesn't have problem with teeth or tooth. This patient can have problem with excessive muscle, some muscle contraction at some distance from point of your pain, point of pain sensation. Sometimes we sense something at distance from real reason, this sensation, real stimulation place. It can evoke misunderstanding, misdiagnostic. So you must understand the principle of pain sensation, pain processing. Perception is your subjective information about some stimulus, where stimulus acts on some receptors, sensory receptors. Not all sensation provoke sense of feeling of perception. You don't pay attention on some weak stimulus with low intensity, but sensation exists in your brain. Sensation is a physical thing. Perception is psychological thing. That's a principal difference. Sensation is physical feeling. Perception uses analysis of information from receptors and your mind. So it influences on subjective identification of stimuli. This rotation, girl rotation, some people can see rotation in clockwise direction. Some people see rotation of this girl in counterwise direction, opposite direction. Can you change rotation of this girl in your mind? And for example, I see rotation in clockwise direction of this girl, but it's subjective. Can you rotate in your imagination this girl into opposite direction? Can you do it? No, it's difficult. Some people can, but now try here. What about here? In my opinion, it's easier to turn switch direction in your mind. It's example of illusion. Illusion and control your perception. Because here brain has more information and some lines, light line, it facilitates your control of perception. So sensory processing, how does it work? At first we have a specific receptor contacting with some stimuli. Usually adequate stimulus evokes excitation of receptors with low intensity. We have specific receptor, sensory receptor. I said in my group that we have two meanings of receptors. Receptors on the membrane, cell membrane, receptors proteins, sensitive to neurotransmitters, hormone, stimuli also, and second meaning of receptor term is special cell. Special cell contacting with stimulus, adequate stimulus, and transform this information to action potential or local potential to special code. Receptor cell. Receptor cell in sensory physiology we can call sensory receptors. To differentiate from membranous. Sensory receptor, special cell. Stimulus, adequate stimulus. For example, what is an adequate stimulus for eye retina photoreceptors, cone and rods? What is an adequate stimulus? Light. What is an adequate stimulus for hearing receptors? Mechanical vibration in cochlea. What is a stimulus for touch receptors in skin? Temperature for temperature receptor, touch, tactile stimulation, mechanical stimulation. So it's adequate stimulus. Can mechanical stimulus evoke photoreceptors? Usually no, but with high intensity, for example, high pressure on eye, can provoke some sensation of flash or beat to eye, flash, but it's not specific. Image not correct in stimulus, mechanical stimulus is not adequate for eye retina photoreceptors. Adequate stimulus with low intensity traction evokes signals in sensory receptors at first local potential, then maybe depends on receptors here on your action potentials, and then propagation signal along sensory pathway to the cortex. Provide analysis. Sensory receptors transforms energy, external stimulus or internal stimulus, External stimulus, physical energy, chemical energy, transforms to signal understandable in the nervous system. Action potential. This process is called encoding, sensory transduction and encoding. Because here we have sensory transduction. Sensory transduction. First step, sensory
Action potential.
transduction. Sensory transduction. It's transformation of physical energy, stimulus into potentials, membrane potentials, local potentials, and then action potentials. On the second step, when local potential transforms to action potentials, sensory receptors should code information. It's called primary encoding. Primary encoding. So, sensory receptors transform physical energy of stimulus or chemical energy into code. Code is a pattern of action potentials. Then this information travels along sensory pathways to cortex, brain cortex.
Brain cortex or thalamus.
So of course, usually very well, sensory pathway includes thalamus, 100%. Sensory pathway including thalamus. Thalamus is the last step before cortex. And then into cortex, brain cortex, hemisphere cortex.
Only olfactory nerve don't go to the thalamus.
Olfactory sensory system has some exceptions, and usually the primary signal is recognized as an ancient cortex. Hippocampal, and entorhinal cortex. This cortex is not visible outside of brain. It's located inside brain. But includes cortex, ancient cortex, archicortex, and paleocortex zones. Brain cortex. During passage of sensory information along sensory pathways, specific or not specific, some information can be changed, but not crucially. It can be amplified or attenuated, increased, decreased, and propagated to different zones of cortex due to divergence. Anyway, it reached brain cortex. Brain cortex is responsible for analysis. Analysis in different zones. It's not a single step, it includes several steps. First step, sensation. Sensation is the physical feeling of something acting on sensory receptor, physical feeling. Sometimes we can't describe this feeling. Your brain responds, but you don't understand that you sense something with low intensity of stimulus. But anyway, brain cortex analyzes stimulus and gives at first sensation, visual sensation. You can differentiate in your mind visual sensation and auditory hearing sensation or touch sensation. And sensation then analyze it again. Higher analysis, brain analysis, and core image. An image you can describe and understand the significance of the image for your life, for your behavior. And perception is processing, higher processing, where perception starts. Perception starts in the cortex. Here we have perception. The term perception may be a little different in psychology, physiology, in some textbook. It can include sensation. Sometimes scientists use term perception to call only higher process with recognition of significance and meaningful patterns. But anyway, we will call everything that happens in brain cortex sensation when you can describe this sensation. Perception, sensation. So perception is based on sensation, but not every sensation can give perception for us. And higher analysis, cognitive analysis. Cognitive analysis is a higher analysis to understand the significance of this image, this sensation for your life, your behavior. Cognitive analysis uses not only sensory processing, but also memory, emotions, a lot of your mind function, higher nervous activity. So sensory processing includes two flows, neural flows. Bottom-up processing, I marked here, from sensory receptors to brain cortex with analysis. It's bottom-up processing, from down to up. And opposite direction, top-down processing. From higher areas of cortex with higher function to lower brain cortex zone areas and under cortex centers to modify perception. So brain cortex image record must be included in the the higher brain area controlling behavior, prefrontal cortex, and we have opposite influencing from higher area, prefrontal cortex, to lower process, and from cortex to sensory pathways, from sensory pathway to receptors. We have feedback for adjustment, tuning of sensory system to perceive information. For example, when you are hungry, you feel better, sense better, smell of food, taste of food. When you are not hungry and maybe you have emotional condition, mental work, preparing, studying, for example, preparing to colloquium, your taste threshold and smell threshold decrease. Where we use sensory information behavior, we start our behavior with information about environment, what happens around you. Adequate information from environment triggers your behavior. For example, question of teacher during colloquium. And after information about passage of your behavior at every step. And finally, information about result, feedback. Again, what is the difference between sensation and perception? Sensation, subjectively, physical feeling, when stimulus acts on sensory receptors, sensory receptor cells. Perception is the central processing of sensory stimulus in a psychological meaningful pattern. Next term, sensory modality. Sensory modality, the quality of your mind. Sensory modality is a sensation. It is not quality of stimulus, energy. It's quality of your perception, sensation, your sensation, where some stimulus act on specific receptor. For example, when light acts on photoreceptors, you feel visual modality. How many modalities we have? A lot. Most people know about 5 senses. Senses and sensory modality a little different. Sensory modality is a complicated sensation. Sense is a more psychological, a result of psychological perception. So, 5 senses: sight, vision, hearing, touch, smell, and taste. 5 basic, but we can sense much more stimulus. Temperature, pain, nociceptive sensation, balance, and acceleration, balance, acceleration, due to gravitation force. What more? Pain, internal kinesthetic sensation, position of your muscle, degree of muscle contraction, and some internal sensation, but more detectable. Primary 5. No? Vision, visual stimulus, hearing, sound stimulus, touch, smell, odor, and taste. Some modalities can have details. They are called submodalities. For example, taste, sensation, taste modality includes salty taste, bitter, sour, sweet, shirin, and umami. Umami is the taste of amino acid. And another, more but fat also, but fat taste modality is not included in many fat textbook. It's new in science. There's actually 6 taste submodalities, 6. 4 main, you must know exactly: salty, sour, bitter, sweet. And 2 adding modalities: umami, amino acid, and fat. Definition of sensory system. Sensory system is a part of nervous system including peripheral receptors and central nerve centers for processing sensory information. First step, sensory transduction in peripheral receptor with primary encoding of information. For example, excitation of photoreceptor in the eye retina on the skin. Next step, transmission of sensory information in brain to brain with sensory pathway. Some pathway can be located in peripheral nervous system. Another part of pathway is located inside central nervous system, mostly in the brain. Next step, sensory decoding and analysis in the cortex with sensation formation. For example, visual cortex located in the occipital lobe, occipital cortex, analyzes lines of object form, of object, here insect, flying insect, um, movement of object, image, color, depth, the dimensions, and form final integrative image, sensation and image. And last step in every sensory system, it works over always is regulation of sensitivity, excitability of the sensory system part in accordance with needs of the body and motivation. For example, this eyes observe, concentrate attention to moving stimulus, clock stimulus, and eyeballs moves. Object form, object information, visual information project to a retina area with the with the greatest visual acuity. How it happens? What about the eye? During lecture, I will give examples mostly about visual systems, the first topic tomorrow. Eye about construction. Retina is located on the back of eyeball. Here, retina. Retina contains several layers. Photoreceptor layers, surprisingly, located on the farther away, farther out of light. I don't know why it happens in all higher animals. Light is propagated across all structure of eyeballs. Corner of the first lens. The first lens in eyeball. It has refractory capacity with refraction. Stimulus. Next, refractory lens located inside the ball lens. It can change direction of light because light is physical optic. And then information about object is projected to retina in opposite, in crossed direction and form image on retina. And retina contains several places. Two main places you must know. One place is called fovea. Fovea contain all the cones. A space around contains rods and cones. More rods on the peripheral part and more cones in the central part. Retina. And second important part of retina, blind spot. Blind spot. What is a blind spot? Blind spot is this place of visual optic nerve. It contains axons of ganglion cells, optic nerve, cranial nerve. It's a place of optic nerve output from retina. So this spot, light spot, doesn't contain photoreceptors. But when we see space around, we don't see blind spots because your brain constructs image on the analysis of information from receptors. This image is artificial. It's not a real image. We can't see a real visual space around us. Everything happens in your mind. Every visual image exists only in your mind. It's based on real physical sensation, but it is not equal physical sensation. So this object, for example, arrow, your retina see in converted image opposite direction with opposite direction, but you see this arrow in right direction because your brain converts, tune out this image again due to perception process. Well, you must know definition of sensory system, what happens inside sensory system. Second, the divisions of sensory system. Each sensory system has three divisions. Peripheral divisions, peripheral part contains receptors, specific receptors. For example, photoreceptors of eye retina. Second division, pathway or pathway, sensory pathway. Usually sensory pathways relay in the lower part of the central nervous system, spinal cord or brain stem, and secondly, its synapses in thalamus. Thalamus arrange, orders information and direct sensory information to specific areas of brain cortex responsible for analysis of this modality. Visual cortex, auditory cortex or hearing cortex, somatosensory cortex analyzes touch, temperature, pain. And third division includes brain cortex, neocortex or archicortex, paleocortex, as an olfactory sensory system. And this picture shows specifics, visual pathway and some branch of non-specific visual pathway, additional visual pathway. The simplified sensory system structure includes peripheral part receptors, sensory pathway, specific and non-specific, and third, brain cortex. Brain cortex area contains more neurons than receptors due to divergence of information from receptors until cortex. Red lines mark bottom-up processing. Blue lines mark top-down processing. Afferent feedback. For example, after eating food, information from stomach travels into brain stem and brain cortex, and brain decreases excitability of taste receptors with afferent axons from brain stem medulla neurons to taste receptors. Tongue, zaman, taste receptors, special sensory cells. It has synapse with afferent neuron, afferent fibers, afferent neuron body, it afferent signal into brain. Medulla. And opposite signal from brain cortex or from stomach to afferent neuron, and afferent neuron controls excitability of sensory receptors. Here are taste receptors. with taste receptors. Afferent information and efferent information. Feedback control. So, sensitivity. Each sensory system varies in accordance with your needs. With details about different parts of sensory system. At first, peripheral part, receptor part. Receptors, sensory receptors, cells or sensory receptor ending have high sensitivity, high excitability to respond to the adequate stimulus with low intensity. There's even low intensity stimulus can excite receptors and change membrane potential. One light particle can change membrane potential in photoreceptors. Only single, only one. Only one odor molecule can evoke olfactory receptor inside nose. Second property, specificity. Its ability to respond to special specific energy of stimulus. Photoreceptors respond to light particle. Hearing auditory receptors, higher cells, respond to mechanical vibration inside cochlea. Touch receptors respond to mechanical stimulus, strengthening, stretching, membrane stretching. This is classification of receptors in accordance with their specificity. The first group includes mechanoreceptors. Stimulation of mechanoreceptors provokes modalities touch, pressure, proprioceptors, muscle receptors, muscle spindles, touch receptors, two touch receptors on picture, auditory receptors here and vestibular. Auditory vestibular, both higher cells are similar. Second group, chemoreceptors. Smell receptors, olfactory and taste receptors. Other chemoreceptors are located in visceral organs and can be sensitive to glucose, for example, carbon dioxide, oxygen. Next group, photoreceptors are responsible to light, rods and cones. Rods longer, cones shorter, smaller. Thermoreceptors. Russian science, usually histology, will teach you about thermoreceptors, Krause, Kolb, and Ruffini bodies. But it's not correct in modern science, it's an old knowledge. Modern science considers thermoreceptors free nerve ending, marked here blue color and red color. A delta fibers usually respond to cold and C fibers to heat. A delta responds usually to cold, free nerve ending, and C engine to heat. Warming stimulus. And the last group, special group, nociceptors. Stimulation of nociceptors, excitation of nociceptors provokes sensation of pain. It's response on damaging stimuli, harmful stimuli. Nociceptor is a high threshold receptors. The rest of receptors are low threshold. Nociceptors are marked as high threshold receptors. Nociceptors responding to damage or high intensity stimulus. So these receptors cannot be excited by low intensity stimulus. they don't respond to usual regular stimulus, but they respond to stimulus with high intensity because they have high pressure. High pressure receptor. High pressure. For example, high pressure, pressure touching with low, with high intensity, cold or heat, very cold stimulus, very heat stimulus, burning stimulus, and some chemical stimulus. For example, acidosis, excessive hydrogen ion and acid inside skin or muscles, lactate, can provoke pain due to this type of receptor chemical stimulus. Histamine can provoke pain, prostaglandin group in inflammation condition. Next property is the ability to respond to a single stimulus with several action potential pattern to provide encoding of information. For what? This property is required to provide amplification of stimulus and to do temporal summation in synapses and provide primary encoding. One stimulus I mark here, a single stimulus, anyway, it provokes primary and sensory receptor agent, several action potential to provide temporal summation in synapses. Because single action potential cannot excite next neuron in the synapse between neurons, and we must have several action potential to propagate signal. And second function, encoding. Because neural system uses binary code like computer. A receptor can respond to stimulus even with slowly increasing in strength, increasing in intensity, due to low accommodation processes. And the last property is sensory adaptation. At the beginning of stimulus action, a receptor sends a lot of action potentials. With long-lasting stimulus action, firing of action, frequency of action potential may be decreased due to sensory adaptation. Degree of adaptation depends on type of receptor and time of stimulus. Another classification of receptors. Most receptors, especially receptors responding to external stimulus, are unimodal, thus more sensitive to a single stimulus emission. Visual receptors, olfactory, taste receptors, and information from these receptors is recognized like a single modality. Visual modality, auditory, sound modality, taste modality. Another group, polymodal receptors. Polymodal receptors acting like this can respond to several energies of stimulus. For example, it contains proteins sensitive to mechanical stretching and evokes mechanical modality, touch or pressure modality or vibration. Next, thermic stimulus to evoke temperature modality to sense cooling or heating, and chemical stimulus. Some chemical stimuli. Most people use cream with menthol, or for example chewing gum with menthol. Menthol is a substance, chemical substance, evoking sense of cooling. But menthol doesn't cool your skin or mucous, mouth mucous. Menthol evokes, excites this type of polymodal receptors, and brain has mistake and recognize this sensation like temperature stimulus, not like a chemical stimulus. It's a mistake. Another example, cooling of skin, ice, line of ice on your hand can provoke sensation not only cooling but pain due to polymodal receptors. Because brain recognizes stimulus in a single pathway due to polymodal receptors responsible to regular stimulus and nociceptive, painful stimulus, damaging stimulus. Next classification, very important for you, primary and secondary. Classification of receptors on primary and secondary is similar histology classification except visual receptors, photoreceptors. Histology consider photoreceptors as primary, physiology as secondary. This classification based on existence of special receptor cells and place of action potential fiber. Primary receptors is a part of afferent ending. This receptor part is located directly on afferent ending. For example, tactile receptors. Primary receptors. Skin. Primary receptors. Afferent neuron, pseudounipolar, usually pseudounipolar except olfactory. Olfactory primary receptor is bipolar. The rest of primary receptors are formed by unipolar, pseudounipolar neuron. Afferent neuron, you know afferent neuron. And dendrite contains some branch. And receptor protein located here. Here receptor. Receptor part. If you remember reflex arc, it starts from receptors and we mark sensory receptor there. Sensory receptor part. It is a part of afferent ending with afferent neuron. Photoreceptor primary. All touch receptors, Pacini corpuscles, for example, here. Muscle receptors, proprioceptors, muscle, spindle, Golgi tendon, organ, fascia receptors, joint receptors. They are located on afferent ending pseudounipolar neuron and olfactory receptor. Olfactory receptor is a bipolar neuron cell, bipolar neuron with shorter dendrite, with receptor field and long axon. Traveling into brain diagram. Next group, secondary receptors. Secondary receptors is a special receptor cell synapsin with afferent neural ending. Primary receptor scheme. And secondary receptor scheme. For example, taste. For dentist student, dental student, more important, taste receptor. Secondary. Taste receptor is a neuroepithelial cell. It is not excitable tissue. It can produce action potential, but it can produce local potential. It sends synapses with afferent ending. Taste The rest, sensory system with secondary receptor, use bipolar neuron. So bipolar neuron in olfactory primary receptors and most of secondary receptors. Visual system photoreceptors, hearing, hair cells, the same vestibular receptors. Next, information about potentials in primary and secondary receptors. Now, this slide, this picture shows some types of primary receptors, free nerve-fading and encapsulated. Sensory transduction in primary receptors. Very simple, very simple. Stimulus, adequate stimulus, connects with sensitive proteins on receptor, membrane receptor. It transforms membrane permeability and provokes change of membrane potential. It's called receptor potential. We can have here receptor potential. The receptor potential is the local graded potential. It cannot be propagated in a stable form, it decays, but receptor distance between place of local potential and place of action potential is short, it's enough. And receptor potential propagated with decay to the first segment, myelinated afferent 18, first Ranvier node. And here, receptor potential is transformed to action potential after providing primary encoding. Receptor potentials are graded potentials. Amplitude of graded potential depends on intensity of stimulus. There's more stimulus, more receptor potential. But action potential is evoked in accordance with all or none law. And receptor cannot code signal with amplitude of action potential, but it can code with frequency of action potential, binary code. We have binary code here. For example, here we have three stimuli with different intensity. Low intensity stimulus, middle intensity stimulus, medium intensity stimulus, and high intensity stimulus. Amplitude of receptor potential changes and becomes more in response to higher intensity stimulus. What happens in first Ranvier node? Amplitude of action potential doesn't change. First stimulus is too low, and response of the first stimulus, local potential, cannot reach threshold. There's lower threshold, and the first case doesn't evoke action potentials, and we don't have sensation. Second stimulus intensity is enough to provoke more receptor potential, and this receptor potential reaches threshold level and evokes some action potentials. And last case with highest stimulus intensity, again, receptor potential reach threshold and evoke a lot of action potentials. And then action potentials are propagated to the central nervous system and to brain. Sensory transduction is secondary receptors. At the beginning, the same. Stimulus provokes a receptor potential. Receptor cells cannot evoke action potential, but it can release neurotransmitter like regular synapse here and evoke another potential like EPSP, but here it's called generator potentials. generated potentials like EPSP, the same, EPSP in chemical synapse, the same, glutamate, for example, neurotransmitter here, and then generated potential is propagated along amyloid enzyme, and like primary potentials, transformed in the first segment of myelinated axon around your node into pattern fraction potentials. Olfactory and olfactory bulb. An olfactory bulb action potential is evoked in the body of olfactory bulb. Olfactory receptors. Olfactory receptor, the drive, body, receptor potential, here, action potential. It's olfactory. Olfactory receptor. Then the information propagated into olfactory bulb. Most receptors respond to stimulus with depolarization, depolarizing receptor potential, except visual. Visual system has some specificity. At first, eye retina is like small brain, neural net. It contains neural networks, network with 5 types of cells. Receptors, bipolar cells, ganglion cells, and 2 inhibitory cells, amacrine cells, horizontal cells. Retina. Details about retina. Photoreceptors, 2 photoreceptors, rods. No, rods. For cones, the same. Rods more sensitive. Photoreceptors, external segment, contain specific visual substance, rhodopsin. Rhodopsin is converted to another form with transformation of opsin molecule. And it provokes receptor potential. What is the receptor potential in rods? Rods or cones photoreceptor are depolarized in the darkness. In the dark condition, photoreceptor are depolarized. In response to lightening, some sodium channel on the membrane photoreceptors become closed. It provokes slight hyperpolarization. Rods as well as cones respond to light with hyperpolarization. The receptor potential in visual system, hyperpolarization. Then information is propagated to bipolar cells. Is this surprisingly? Bipolar cells don't produce action potential in this form, again, with local potential. But some bipolar cells respond with depolarization due to special receptors to glutamate, neurotransmitter photoreceptors. A special receptor transforms hyperpolarization to depolarization. These bipolar cells called on cells, to on. Next type of bipolar cells called on transduct again hyperpolarization. Then bipolar cells ganglion cells. Synapse with ganglion cells. Ganglion cells like the first afferent neuron. Synapse, second synapse. Big cells. And on the first channel, ganglion cell responds to signal in synapse with action potential. Second case of channel, ganglion cells responds to light with inhibition. And decrease action potential. Usually ganglion cell has a regular background activity. With inside on channel, ganglion cell respond to lightening of receptors with more action potential on response. Another time with decreased frequency of action potential inhibition of response. It's a principle of visual recognition. Another type of cells in retina, horizontal cells. Horizontal cells controls synapse, first synapse in retina between photoreceptors and bipolar cells. Amacrine cells controls second synapse. Both cells are inhibitory cells to provide lateral inhibition in retina. More contrast, more detailed lateral inhibition. We studied lateral inhibition in the previous colloquium. Put here a scheme of signals inside retina in vertical column. Hyperpolarization receptors, two possible responses in bipolar cells, on response, off response, and a response in ganglion cells. Axon of ganglion cells form optic nerve and leave retina in a blind spot place. Next, classification receptors on the base of adaptation, ability to adapt, to be adapted. This picture shows stimulus time, stimulus duration, turn on, turn off, for example, mechanical stimulation, and we can have two principal responses. Signal can be perceived in receptors only at the beginning of stimulus, here, and then receptor doesn't respond to stimulus. This group is called rapidly adapting receptors. The main example, Pacini-Karpasa, the receptor of skin responding to vibration. And most receptors are slowly adapting. They maintain receptor potential and then action potential during all time of stimulus, but this receptor can decrease amplitude of receptor potential and frequency of action potentials at the end of stimulus, less and less. So after dressing immediately, we can sense your dress, your clothes. Your clothes contact your clothes with your skin, but then you don't pay attention to your clothes because your mechanical receptor on skin are adapted to clothes pressure of your clothes on skin. Visual receptor, hearing receptors, most receptors are slowly adapting, but inside this group, we can divide this group more adapting slower or faster. The most slowly adapting receptors are nociceptors. Adaptation to painful stimulation mostly is controlled in the central nervous system, in the nerve center, but not on the receptor level. one afferent neuron has a lot of branch. So stimulation of each branch provokes excitation of this afferent neuron, and all receptor ending of a single sensory neuron is called a receptive field. Receptive field for black neuron, receptive field for gray neuron. Bipolar cell can have synapses with many photoreceptors or only with a single photoreceptors. The size of receptor field may be different in different zones of receptor area or in different sensory systems. Receptive field may overlap the secondary order neuron pathway, sensory neuron and spinal cord receive signal from several afferent neurons. Each afferent neuron has a proper receptive field, and you can see that the receptive field of this third neuron overlap. The sensory pathway. The sensory pathway includes afferent neuron, relay interneuron in lower part, central nervous system, spinal cord, brainstem, higher part, thalamus, and other centers of brain. Usually, each sensory system has several pathways, faster and slower pathways. Sensory pathway has two principal functions: propagation, passage of sensory information from receptors to brain cortex, and second function, transformation of signal. Sensory pathway change signal. It can decode and again encode, recoding, provide a recoding of signal, change of intensity of signal, increase or decrease, and provide more contrast. Pay attention to more important sign of stimulus, more important details of stimulus. Sensory pathway, each sensory pathway has several levels, synaptic relays, and many channels for passing information. And the sensory pathway has convergence and divergence inside sensory pathway, and usually form some funnels. Convergent funnels due to convergence of information from several receptors to a single afferent neuron. Here are ganglions in visual sensory system. And later, sensory pathway has a lot of divergence to propagate information in different brain areas responsible for different functions. For example, visual stimulus propagates to several brain areas. Each brain area is responsible for some analysis of some details. About visual system, very important part. Eyeball, right, left, retina. Retina optic neuron, it has partial chiasm, partial crossing fibers, optic tract, thalamus nucleus, I will hold it in later, and then the cortex. Specific visual pathway. Visual pathway projected brain cortex. Brain cortex zone. Now you know motor cortex, precentral gyrus, postcentral gyrus, primary motor cortex, second column.
premotor cortex, prefrontal cortex, the most important to control behavior, prefrontal cortex, postcentral gyrus, somatosensory cortex, primary somatosensory cortex, mammary body, and occipital lobe contain visual systems. Primary visual cortex, V1, secondary visual cortex, 2, third visual cortex, associative area, temporal and parietal associative area. Everything here, visual cortex with different zones. V1, primary, secondary, third, tertiary, and associative. There's a lot of areas responsible for different functions.
Due to divergence further. Degree of convergence funnel may depend on receptive fields. Retina has some specificities in accordance with color vision and black-white vision and function of cone and rods. Fovea area contains just cones. And here you can see bipolar neuron, ganglion neuron. Receptive field is very, very small. It contains only single cone, single receptor. Parafovea area contains mostly cones, a few rods, and here we have convergence of several receptors to a single bipolar neuron or ganglion neuron, but convergence degree is small. And peripheral part of retina contains a lot of rods. Usually, totally, retina contains 90 millions of rods, 90 millions in retina rods, and 4.5 million cones. This cones, a number of cones, few can compare with rods. Rods is much more. And peripheral part of retina has a high degree of convergence, high convergence. A lot of rods converge signal, send signal to a single bipolar neuron and a single ganglion cell. What is the advantage and disadvantages? In rod, black-white vision, rod are more sensitive neurons. They can respond on low intensity of lighting, low lighting. With low lighting, we can excite each rod, but single signal from one rod, not enough to excite bipolar neuron. But we have high convergence. And signal from each rod is summed due to spatial summation on bipolar cell or ganglion cell. Spatial summation provokes response of pathway neurons, afferent neurons. So low intensity lighting due to large size of receptor fields and spatial summation evokes response. In cones, each cone can respond, but due to absence of spatial summation, signal cannot be propagated in synapses between cone and bipolar neuron, ganglion neuron. So we don't have sensation. There's more rod vision is more sensitive to low intensity lighting. So in a dark condition, rods are attenuated and increase excitability to see light. Fovea area, another case with high intensity of lighting, high lighting, daytime, or electrical light. Again, in cones and rods, we have response, but more response. So each cone can propagate signal into brain. Can the propagated signal to brain cortex, and brain cortex can form a sensation. So we can differ to light point due to a narrow, small receptive field. And fovea area is the area highest, greatest visual acuity. Greatest, it has greatest visual acuity. Visual acuity, what does it mean? You can recognize well object. Actually, only a small part of your retina can see object with a lot of details. See well, but you think that you see everything well. Why? Because your brain construct information, integrate information from fovea, and your eyeball are moving all time. In at rest, when you see on something, look on something, look at an object, your eyeball are moving with a slow movement. When you want to see, to look, to recognize some interesting object, your ball move faster. Special control. For what? To provide projection of more important visual object. For example, face of your partner when you are speaking, talking with somebody, face or eyes or mouth to detect speaking, project this important, significant image exactly to fovea your mouth. What's this? So, cone vision has advantage, greater visual acuity, better visual acuity. But it doesn't work with low lighting conditions. So with darkness, brain change cone vision to rod vision. I will check presence at the end, so a little time is left. And later inhibition in retina provides more contrast, and we have more details in details of object contour. The classification of pathway in accordance with the speed of propagation and amount and numbers of relays, synaptic relays, and functions. Specific and non-specific pathway. What is the difference? Specific pathway are specific for each sensory system and transmit information usually about only a single modality. Non-specific pathway can combine, integrate signal from different sensory systems and transmit information about several modalities. For example, temperature and touch together, in pain together. You can see the difference between them. If you remember, ascending pathway from previous colloquiums, ascending somatosensory pathway, lemniscus pathway here, lemniscus pathway, first neuron, peripheral neuron, second neuron in spinal cord or spinal cord, spinal thalamic tract, or lemniscus pathway, brain stem, and third neuron in thalamus. Third neuron in thalamus, and then to cortex. Non-specific pathway has a lot of synaptic relays. We can't call exactly levels of synaptic relays. Specific pathway is propagated across specific nucleus of thalamus. Non-specific pathway through non-specific nucleus. A specific pathway is projected to specific projective area, visual area or auditory area. Non-specific pathway is projected to associative area. This is table you can read in my video lecture presentation. I will not now read it again. The central visual pathway. Pay attention to partial chiasm. Partial chiasm. Signal from nasal area of retina, crossed out to another part of the retina. Signal from temporal area of retina passes to the same side of brain. Partial chiasm. 60% of fibers in optic nerve cross out. After optic chiasm, nerve is called optic tract. Optic tract is projected to specific thalamic nucleus, lateral geniculate body, lateral geniculate body, and then is propagated directly to visual cortex. Branches of optic tract project forms non-specific pathway and project to superior collicules to provide, for example, orientation reflex to new stimulus. This orientation reflex, and turn your head to new stimulus. To protect, to provide some reflexes, adjust vision and eyeball to recognize stimulus, pupillary light reflex, and accommodation reflex. I will ask reflex are pupillary light reflex and accommodation reflex. Reflex are. Tomorrow control work will include reflex arm. Pay attention. It's a main medicine diagnostic test to understand the condition of central nervous system. Pupillary reflex arm. Pupil is a small hole. Is a place of light. Size of pupil may be decreased or increased in accordance with light. With high lightening, pupil decreases in size. Constriction, half constriction. In dark, pupil dilates. Pupil reflex, pupillar lightening reflex is constrictor reflex with parasympathetic FM fibers. Eyeball, pupil, light. Retina, photoreceptors, bipolar cells, ganglion cells, optic nerve, then chiasm, optic tract, then pretectal area, pretectal nuclei, first relay, synaptic relay, then third autonomic nucleus, Ezekiel best 5 nucleus is autonomic nucleus,
parasympathetic nucleus, in brainstem, the brain, and afferent pathway, long preganglionic fiber, synapses inside ciliary ganglion, and then ciliary ganglion neuron innervate parasympathetic neuron innervates muscle constriction. Pupil constriction. Neurotransmitter acetylcholine, muscarinic acetylcholine receptor, and blockade muscarinic receptor with atropine, dilate pupil. For example, ophthalmologist uses response with atropine drops temporarily dilate your pupil to see better your retina with light. Accommodation methods. Accommodation. What is accommodation? Accommodation is a change of lens diameter with looking at closer or farther away. What happens? At distance. At distance, we have direct path, direct light emission, and refraction is not high. Refraction to project forward. With closer object, light rays enter with more angle. It's necessary to increase diameter lens, to increase refractory capacity, and again provide projection of object to fovea. It's called accommodation. Accommodation is increase lens diameter height. However, how it happens? It happens with constriction of ciliary muscle. Ciliary muscle. Ciliary muscle constricts by parasympathetic fluids. With constriction ciliary muscle, it becomes flatter. It really it becomes more how to say more shape. It facilitates tendon surrounding lens, and lens becomes more thick, with more diameter. It's accommodation reflex. Why it happens? When it happens? Accommodation reflex. Eyeball, light, lens, light. Again, optic nerve, optic tract, thalamus, lateral geniculate body, cortex, cortex, and then after cortex, again, pretectal area, Edinger best 5, ciliary ganglion, and then ciliary muscle. Pay attention that accommodation reflex,
accommodation reflex travels across cortex when your brain turns on accommodation reflex. When you can see well text or space around you, your brain turns on accommodation reflex. So this reflex R includes cortex. Well, I give here pupil light reflex or pupillary light reflex pathway, but you must study yourself accommodation reflex in textbook, and I will explain during class. Well, study yourself brain cortex because I don't have time, and check presence. Алама. Амели. Алиф Москвы. Банакшенко. Игорь. Писаров. Барман Фарид. Гафур Хаят. Дашкаев. Дада Килех. Джуди. Заравшани. Махмуди. Маазами. Мухаммади. Мутуме. Эльхати. Масуди. Well, second. Зилаев.
Here's this. Here's this.
Wait. Mahmud. Nabipur. Ali Ayselek. Rad Manish. Disappear. Rahim. Rahmat. Rashid. Rahmatiyan. Dismanipur. Sophie. Farfazi. Sipati. Manipur. Hossein Jabir.
Yes.
Shahbaz. Shayed Bahar. Zahra. Muhammad Azadeh. You can be free. Bye.